Hi.
Hello. Hi, good evening. I'm so happy to see so many people join tonight. It's cold and wintry here, but Evelyne is joining us from Moray Island, where it's 7 a.m. and it's super sunny. I'm going to ask her to give us a little view of her office in a minute. But just to introduce Evelyne, she's head of reef restoration and science for Coral Gardeners. And Coral Gardeners is an NGO working on restoring coral reefs around Moray Island in French Polynesia. And just the work they're doing is so beautiful, so stunning, and full of positivity. I thought I couldn't resist asking Evelyne to give this seminar this evening, and I thought it might brighten your moods as we're entering wintry February. So Evelyne, over to you. Can you please show us your office again?
All right. So, thank you for inviting me. So, yeah, as Cecile said, it's quite early for us in the morning. It's only 7:30, but that's the view of my office. I don't know if you can see it. It's quite nice and very sunny. The sun is out now. And that's the bay. And over there, when it's the whale season, we can see actually whales. So right from our office. And that is our little boat that we use to go to our coral reef and coral nurseries. So yeah, welcome.
I'll let you share your screen.
All right, so I will just share basically what we are doing here in Coral Gardeners. Okay, so if...
Super, c'est bon.
Yep. So here we go. So I don't know if you can see. You can see my screen?
Yeah, c'est bon.
Yeah, c'est bon. All right, so yeah, Coral Gardeners were in Moira in the French Polynesia, so it's in the middle of the Pacific. And we are doing coral restoration around Moria to start with. So first, before I introduce me a little bit. So I'm Evelyne. I have a master in marine biology in Marseille in France. So yes, I'm French. And I spent the last 11 years in the Maldives doing coral restoration along resorts. And as well, I was a marine biologist for the resort, dive instructor also, and I spent also lots of time educating staff and guests alike on the marine life of how beautiful it is, why we need to protect it, and how we can protect it, and what people actually can do. It's very few steps that I think all of you may know. And I joined here back in September 2020, so it's been 5 months, and it's been a very nice ride, and I basically love it every single day because working with that is just amazing. So basically who we were, so it started with Titouan, the founder. So we all surfers, marine life lovers, free divers, fishermen, math scientists, engineer, who love the reef. And one day when he was surfing a few years ago, about 5 years ago, he saw here's the coral reef, all white. So he asked why it was white. And he realized that because the coral were dying. So he wanted to do something for the reef. That's why he created Coral Gardeners. And just because you are far away and in London, I just wanted to show you a small video to brighten up your evening. So that's what we usually see when when we go out, and it's the whale season. So that's a mother and its calf. So now we are a team of 20 people. Was working full time, saving the ocean and trying to find a new innovative way of doing coral restoration. So basically our mission at the moment is to change the way we see coral restoration, is to try to create new technique, new method, new technology to make the coral restoration easier, faster and better, with better results. Because in the end, what we want is a world where the reef no longer needs us. So that's the aim we want to have. So to restore the reef, help the reef so that it doesn't need us anymore. So I don't know how much you knew about coral reef, so I made a quick recap of what is a coral and what is a coral reef, just so we all know what we're talking about. So coral reef, you might know they're made of coral mainly. They're the reef builders. So corals, they're animals. Polyp, as you can see on the right side. So they're a family of cnidarian. That means stinging cells, so like jellyfish and sea anemone. And they host zooxanthellae, microalgae. That gives them the colors. That's why they're pink, blue, brown, sometimes green. And they give also the food by photosynthesis. So about 90% of the nutrient is given by this zooxanthellae. And the 10 remaining percent, the polyps catch them with a tentacle at night. And you can see it takes a long time to build a reef, because the polyp is actually taking the calcium and the carbonate in the water to build the exoskeleton. So it takes a year to grow a centimeter, sometimes to 20 to 30 centimeters, depending if it's a branching one or a massive coral. So yeah, it takes usually millennia for coral to build a coral reef. So it takes quite a long time. And then coral, you might know that the coral reefs, they cover less than 1% of the ocean floor. Basically it's 0.1% of the ocean floor, but it's home to 25% of all marine life, because they need it for shelter, for food, or for reproduction. So if there is no coral reef, we mostly lose 25% of the marine life. But at the moment, 25% of coral reef are dead, 50% are not in a good health, and only 25% of the world coral reef are actually healthy. So there's not much left for us to see. So there's some threat of coral reef. You may know there's some global threat and some local threat of the coral reef. So the famous one, the ocean warming. Sea level is rising, so when the ocean get warmer, the coral tend to die, because they only have a range of life between 23 degrees to 29 degrees. Hotter than that, they usually don't like, and they might die. The sea level rising, well, if it rises too fast, they might not be able to protect the island. Acidification of the sea, that's also another major issue, is because all the CO2, like a quarter of the CO2 is trapped in the ocean. And so it bound with a carbonate, so it make it unavailable for coral, but also crabs or anything to actually build the skeleton. So that's make the coral weaker and easier to catch any disease or break. And of course, the most famous one, El Nino oscillation, that change the weather pattern. So it may be drought, it may be flood, sea temperature will rise also. So all of that creates threats to coral reef, and they actually don't have time to make it better. So there is, so other than the coral, the global threat, there is a local threat, so that the one we're working on here, because trying to make the sea cooler is not something we can do at here, but we can work on overfishing. That's another threat of the reef. Tourism also, bad tourism, so people stepping on the reef, anchoring on the reef, that destroys the reef. Also, and whatever when they're building, reclaiming, also islands that destroys the reef, and hence it tends to die. So that the major threat to coral reef at the moment. Another threat, why it doesn't work? Oh, here you go. So why do we need the reef? Basically, they are quite important, as I say, because they're home to 25% of marine life. But for us, why should we care? Because they protect island. They're massive buffer from the wave action. So here you can see it's very calm and quiet. But if we go a little bit further, it's actually the ocean is quite big, so there is nice wave. Everybody's surfing. But they also give income by tourism. They give food also with fisheries. So they actually benefit people, for 500 million people actually around the world need the reef to live, for food or income, and then generate more than $370 billion per year. So that's kind of why we need to protect the reef. So here, what we're doing in Coral Gardeners is we try to restore the reef. Moharia is not in a very bad shape, but if we don't do nothing, it will go very bad. So first, what we started with Titouan, it's basically they were collecting corals of opportunity. So this kind of corals are fragments, broken fragments that were found in the sand. So they were broken because either the fish break them to find the crabs and small fish living inside them, or people, snorkelers, or divers stepping on coral. But anyhow, collecting corals, and the first thing they were doing is picking up bamboos in the forest. With cutting the bamboos in small sticks and gluing the coral on the stick, and actually making a hole in the dead coral block, as you can see on the picture, and inserting the bamboo in there and letting the coral grow. So with that technique, they planted about 10,000 coral all around Moria. But after a couple of years, we realized that it didn't really work because the bamboo were falling off, the fish were coming and trying to eat them, and so coral were falling off. They were not really attached. It was not a huge success rate. So they found out they had to glue the coral on top of the coral blocks. So instead of going to the bamboo to glue the coral in the bamboo, then drill a hole, put the bamboo and glue them, they just took the coral fragment and cement them straight on the coral blocks. So that's what we did for the remaining about 5,000 coral. And I will show you two small experiments that started a year and a half ago. So one is right here on the reef in the Cook's Bay. So as you can see on the picture, so we planted about 90 corals on 20 square meters. So we had plots of 1 square meter, so we had 20 plots, and one every plot planted coral. Sadly, 2 days after they planted the coral, there was a massive flood and big storm, so we lost most of the coral. But the one remaining, they're about so 40% of the survival rate from the first 90 fragment planted. So it's not the best survival rate in the world, but weirdly, the one that survived, they just grew like they exploded. So from the coral, you can see they had like 2 branches each, and now after a year, they're just like massive. So we don't really know why, what happened, because here the bay, it's a river coming in. So when there is flood, there is like mud, sediment, everything. So we don't know actually how they can survive, and they survive very well. So that's another study we're gonna do this year. Then second one is more on the north side of the island. It's on the MPA. So then again, we cemented more than 300 fragments on 32 coral blocks or boomies. And we just again try to see monitors for the survival rate, and we found out that they survive much better than on Cook's Bay. They're more than 60%, 68% of survival rate, so it's quite good after a year and a half. But weirdly, they didn't grow as fast as the other ones. So again, we don't really know why. Maybe because there's more predation, maybe because there's more wave action, so they tend to enclose and attach themselves more than actually trying to grow. So that's interesting to see these two different kind of study. But then I arrived and we started a new method with super corals, rock nurseries, and then transplanted corals back on the reef. So super coral, basically that's, we love that name because it's catchy. For us, the super coral is a coral that has survived bleaching event. So last year, the team, when it was bleaching event, they went on a survey on the reef and they took a GPS coordinate of all the coral that didn't bleach and survived this bleaching event. So this year we came back and we pick up some fragments of coral. So that's the technique, pick up like some 10% maximum of the colony. Then insert it on the ropes, wait that it grows. When it grows big enough, we trim it. And this fragment, we cement them back onto the reef. And then hopefully they will spawn and make more babies. So that's basically what we did. So here is us building the nursery. So it's in 2 meters of water. It's quite shallow, but they're doing quite okay. So that's very good to see, because if the coral can survive in 2 meters of water, they can survive basically anywhere. As you can see, we fragmented some coral, not much, and whatever fragment were not small enough. So we wanted the fragment to be about 3-4 centimeters long on the rope. So we were refragmenting them on the boat, trying to keep the coral as much as possible in the water. insert them on a rope. Once the rope was done, it was hanging on the boats until we could actually attach them on this nursery. So in this place, there are 3,580 fragments of coral from 146 colonies, 20 species of them, 9 genera and 20 species of coral. So we try to make as much diversity as possible to avoid like genotype problem and bottleneck issue when we're gonna transplant back. So when we're cementing, the same. So we're gonna make a plot or we're gonna shoot the boomies. We usually cement depending on the size of the fragment. That will be 8 fragments per square meter back on the reef. And of course, that will be easy just like this, but we do some monitoring. So we monitor the nursery for health, bleaching, growth, predator, disease, and we take some photos. So health and bleaching is 1 to 4. So 1 it's 100% alive, 4 is 100% dead. 2 is 50%, more than 50% alive, and 3 is less than 50% alive. Bleaching the same. 1 is super colorful, 4 is dead, 2 and 3 is in the middle. We check for predator if there is any disease, and this we do it for all the fragment on the nurseries. And the growth, we picked up 500 fragments, well, completely random selected, thanks Excel. And so we take the growth, so we take the 3 measurements to calculate the ecological volume, so the maximum height, length, and width of this coral, and also we take a picture of them so we can see how they grow and how fast they grow. So that we'll do for the nursery. We will do also the same for the transplanted site, and we do that every 3 months. Unless we are on a bleaching alert. So at the moment in Moorea, we are in bleaching alert because we might have a bleaching event in a couple of months. So we are doing a health monitoring every month. And if it occurs, if the water is becoming too warm, we might cover the nursery to avoid too much sun, try to protect the fragment as much as possible. And then every year, so when we're going to transplant coral, just before that, we will do a survey for corals, fish abundance, recruits, and inverts. And then we'll do it again right after the transplant, and then every year, both in the transplanted side and the control side. So we see if actually our work is working or not. So hopefully it will work. So we're mainly in the water doing that, but sometimes when we go out of the water, we love to educate and to talk and to tell people that we need to protect the reef. And we do that in school, in projects. So we have a beautiful project with a school here, because as you may know, with COVID, we can't, even if there is not much restriction here, but we can't really go into school anymore. But we do a beautiful project with one school here with 2 classes. So they're doing mini coral restoration project. So they cemented some fragments back on a reef, and they put also some fragment on the rope. And they monitor all of that. So we told them how to recognize coral, the health of the coral, if coral is bleached or not, and how to measure them so they can see the growth. And that's a full year program. So in the mathematics, they learn the formula to calculate the volume. They also all speak in Tahitian because there is a lady explaining the culture and the heritage link to the sea, to the mountain, to the island, to people. So they have all-in-one project, which is super cool because they spend like once every 2 months a day in the water, so they love it. So we used to go to school also quite a lot to explain and to teach them about coral reef and the importance of protecting them. So beside the school, we did also, but we stopped again because of COVID, some eco tours. So tourists, people could come once a week or twice a week here for half a day. Again, we explain what is a coral, what is a reef, the need to protect, we educate them, and then they were going right here in the lagoon and planting coral. We do also some talk, like me here, but Titouan, here is Titouan, the founder, did some talk also in France, here with the CRIOB for the Science Day in Tahiti also. So we do a lot of talk, but now also again with COVID it's less, or it's by video. And also we try to raise awareness, and that's how we get our fund majority is with adopt a coral. So people can go on our website, help us by adopting a coral and supporting us. We also receive donation, and we work with some partners. That's how we actually found the organization. And also the last but not the least, we have media ambassador, because you may or may not have seen us on social media. So all these people and even more our ambassador, and they just talk about us and work with us, come to visit us time to times. And they're sharing our work. So I think that's it. I try to explain as much as possible. So I don't know if you have any questions.
Merci, thanks Evelyne, that's beautiful. I'll just open up the room to anyone who has questions. If you want to put on your camera, please do so. It's nicer to see you.
Yeah.
Hi Evelyne, I've got a question if that's okay.
Yeah.
Yes, sure. Hi,
thanks for your talk. That was really interesting. And oh my God, your office looks beautiful. I'm so jealous compared to you right now. I just wanted to ask, so I work on coral reefs as well for my PhD, and I study reefs in the Indian Ocean, not in the Pacific, but quite similarly, I focus on corals and what lives on coral reefs as well. And my question is, have you done any surveys in your local reef area about why the local reefs are dying? You know, the causes of local mortality, is it just bleaching led, or are there other maybe more different reasons like sedimentation? And it seems that from your pictures, there's a lot of bacterial mats and algal turfs around where you have your new coral colonies. And so you can expect the conditions to be a bit anoxic. And so how are you trying to mitigate that? Are you looking for maybe deeper sites to plant your coral colonies away from the bleaching areas? That's it.
Cool. Right, so I'm not aware of much study here that has been done. But yes, we know that they died because of the global threat, but mainly there are some rivers, and when it's raining, it's flooding here. So all the sedimentation, all the from the runoff from the agricultural fields and all that goes into the lagoon. So that didn't help, because now, as you could see, there is lots of algae growing. That they were there before, but they increased like a lot. So we are talking with people on the island to mitigate this. That takes quite a long time. But weirdly, where we have like this cyanobacteria and algae, it was just 1 day, 2 days. Then because where the nursery is placed is next to a channel, so there is some current passing by, and sometimes it's quite a lot of current. So that's probably how they're doing. They've been there for 2 months, and we still have more than 95% of survival. So I'm very much impressed because it's very shallow.
Yeah, that's super impressive. You can imagine that if you're expecting.
I hope I answered your question.
Yeah, that was great. That was super great. You can imagine with the bleaching event coming that it might turn a bit hot in those sort of shallow areas. Have you looked at the slightly deeper reefs around the island and whether maybe there's less bleaching happening there?
Can you hear us, Evelyne? Oops. Oh no, it sounds like we've lost Evelyne. Oh, can you hear us?
Yes, I can. I just disappeared for some reason. I don't know.
Okay, it's back. You're back. It's fine. Margot, do you want to ask your question again?
Yeah, sure. I was just saying, if you're expecting a bleaching event in the next couple of months, have you looked at maybe some reefs around your island where the bleaching might have a cutoff? So maybe somewhere which is slightly deeper but still light enough to have coral growth that maybe you might be able to do your transplantations into?
Yeah, the weird thing is here. I was talking with a CRIOP, which is like a French Institute for the Sea, and they were telling me like the last bleaching event, the drop off of the island was much more impacted than the coral in the lagoon, because there were a much more bigger difference in temperature outside than inside in the lagoon. So that was very weird for me. But we don't have any area because here we work with permits, and the permits that we got is for this area only, and it's quite shallow. So the only thing we can do to mitigate the bleaching event is to cover it with shadows. And so we're gonna shade it. Sadly, that's the only thing I can do.
About the shading, I had a question. I was wondering, you said you could shade the nurseries, but then I imagine the transplant areas are much bigger, right?
Yes, a transplant area, we can't do anything. But we will transplant, it takes a year. So we put coral back in November, December, so they will be able to be transplanted next year, maybe in January, December, January. So, but once they will be on the reef, yeah, we cannot do anything. It's too big.
Does anyone else have a question? Yeah, go ahead.
Not a question, but just a hello from one of your Coral Gardeners collective members. When I saw that you're presenting today, I was like, oh my gosh, I can't believe they're here. So yeah, sending love.
Thank you. Anyone else?
I just wanted to ask a quick question about the social media side of things, because I first came across you guys because there was a really popular video on Instagram which sort of talked about the coral reef being the ocean's lungs, and it was shared, lots of my friends shared that. And I just wanted to ask you, what do you think gave that campaign like so much cut through, and did you see a big increase in support after that?
We did see a big increase in coral adoption after that. We still don't really know why, but we probably posted at the right time and the right day, and it got shared by millions of people. But then, yeah, we did see a very big increase. So thank you.
That's good to hear.
Well, about that also, I was wondering what your main sources of funding are. So is it I think you were saying it's adopting corals, right?
Yes, it's adopting coral, our main source. Then also donation, people, they donate, and partnerships. So we work with some brand as a partnership. So that's the main three source of funding that we get.
Can I invite other questions?
I had another one, if that was okay. I know I've already asked two, if that's okay. How many of all of the corals that you've grown, how many of them do you think have survived since the start of the project?
Since the start of the project, for the 15,000, they're all around the island. I haven't checked yet all of them, so they're very far. But looking at the picture of everything, I would say the average is 70%. 70, yeah.
That's really awesome. Is there like a difference between the different like groups of corals, like different types of corals that are surviving better than others? Because we see that in the Indian Ocean, where the acropora corals, the one that make tables, or very thin branching corals, have died off a lot more in bleaching events than sort of more Porites, sort of massive like corals. So I wonder if you've seen something similar.
Yeah, similar. Yeah, the one naturally the one we find are Porites and Polynesia mainly. Acroporas, as you said, like they tend to decline. But weirdly, they're doing actually very good on the transplanting area. Once they actually like glue themselves, they're okay. So yeah, that's mainly here. They've worked on Montipora and Acroporas to transplant them, and Pavonas. They love Pavona, and it's everywhere. It's very nice. So, and where do you work in the Indian Ocean?
So I work in the Chagos Archipelago, so that's pretty much in the middle of the Indian Ocean, so like underneath the Maldives, pretty much.
Yeah.
It's like similar to the Maldives. Yeah, and there was a very big bleaching event there in 2015, 2016, where up to like 70% of the corals died. So any research on like recruitment and things like that is brilliant. It's really nice to see somebody like actually do something about it. It's great.
We think that it's nice to do research, but we actually do it. We actually have to do something now. So we're trying our best.
We have a question from Emily. She said, hello, I might have missed this, but could I ask how you select which coral colonies to take cuttings from? Yeah, I think that's about the supercorals then.
Yeah, that's our definition of supercoral. So we selected like stress tolerant, heat tolerant coral. By actually last year there was a bleaching event, and even the year before. So the guys last year, they went with a GPS and they marked all the colonies that didn't suffer for bleaching event. Well, they could see, like they didn't go all around the island because that's a lot of things, but enough for us now to go back to these colonies and fragment no more than 10% of that colonies and putting them on the rope with the hope of these guys will be our super coral.
And I guess Emily's question is about whether you manage to get enough of diversity in the coral species you get.
Yeah, and the diversity, we try our best. So as I said, it's 146 colonies and 20 species out of that. So we try our best to, in each species, to have at least 5 different colonies. So we can increase the diversity.
Hi, can I just jump in and ask a question, please?
Sure.
Yeah, just kind of following on from that point. I suppose if you select species which are already quite tolerant to thermal stress, there's potential there for you to kind of go down a kind of functional bottleneck where you just end up with species that are already quite resistant to thermal stress, and I guess you lose the species which are quite at risk already. Is there any work going on to try and target these at-risk species and improve their capacity to tolerate thermal stresses?
There are some studies. It's actually quite hard. There is a couple of PhD going on, candidates here. But around the world, it's harder because, you know, the corals, they live with a microalgae, the zooxanthellae. And that's this zooxanthellae, once the temperature is too high, instead of giving nutrient, they give toxic to the polyp. So one study, they try to make zooxanthellae resistant to bleaching. So they actually don't make any toxin. But the polyp, the coral didn't like them, and they just release them. So we can't do anything. And I know there's some, I think in Hawaii, or in the States at least, they're trying to modify some genes of the polyps to make them more resilient to the heat. But once again, we don't know because it's a symbiont. So we don't know if this coral, when they will have like a new gene, like more heat tolerant, it's which algae they will choose, because at the end of the day, they choose which microalgae they will go into them. So that's quite hard.
Yeah, that does sound very difficult. Thanks for the answer.
Yeah, you're welcome.
We have a question from Connie McDermott also about, so at several levels. First, do you have support from higher level government or local government? And then also, how interested is the local community? Because of course you were saying overfishing is one of the biggest threats. How involved are the local community, and how are you getting them involved?
So local government on the island, yes, we do have some support. They like what we're doing, they like our work. Higher level of government, not that much, not as what we would like, because they just don't have time. And the local community, they are quite encouraging us. We have some very good feedback when we go and talk to people. So yeah, they're trying to slowly get in the mind that we need to protect the reef, and they are protecting like Moria. There's lots of MPA around the island, and that we call Rahui here. So that's what they used to do also, to protect a part of the lagoon for a year or 6 months, and then another part of the lagoon. And so they used to work with that. So locally, yes, we are supported. The higher level of the government, not as much as we want.
Thank you.
Hello?
Yeah, you're in and out, but it's okay, we still have you.
Okay.
Could I invite more questions? I think, Evelyne, if you have, we'll send your contact details if people want to come. I guess, are you, I guess you would be open to having PhD students, or when we're back out of lockdown, having master students come to visit you. So yeah, it'd be nice to have, to set up a partnership and a relationship around that.
Yeah. Ah, thank you, Alistan.
Oh, that's really nice. Yes, so yeah, please do adopt corals.
Thank you.
And we'll include your details on the Biodiversity Network website, and we'll make sure we keep a relationship going.
Yeah, definitely. Yeah, let's do that.
if we don't have any more questions, we'll close for the evening. Everyone's in a dark February winter, so I think people are getting tired. Have a great day. I know you're going to monitor your corals right now, right?
Yes, that's what we're gonna do. Like, the team is here, everybody is here. Yeah, they're waiting for the go, and yeah, we'll go in the water now.
Enjoy. And thank you. Thanks so much.
Thank you.
Bye. À bientôt.
Bye. À bientôt. Thank you very much.